Yes, good morning students. Today in next class we will start about vegetative propagation. Today we will take the two topic vegetative propagation part 2 and then asexual mode of reproduction in lower organisms. So in last class we have to study and we have to discuss about the vegetative propagation and then types. I have to explain two type of vegetative propagation. First is a natural, second is a artificial. Natural vegetative propagation are three types, by stem, by root and by leaf. And then artificial vegetative propagation in which human interference involvement is the three types: cutting, layering and then grafting. So today we will discuss about the cutting, layering and then grafting with the example and with the process. So first we take the artificial vegetative reproduction part first by the stem cutting. As we know, the word cutting means cut. Clear? So in this process we cut the small piece of a stem and then grow the new plant. That time the process is called artificial vegetative propagation by stem cutting. Clear? If we discuss about these cutting method, we have to know about following points. First, the propagules. In terminology class I explain about the propagules. What is the propagules? Propagules are the plant parts which use in the vegetative propagation, like stem, root and then leaf are propagules. So in case of these, propagules are a small piece of a stem. That means the long stem, we can take the small piece of a stem and the size, the minimum size of a stem 20 to 30 centimeter long. It is essential for the experiment that we have to take the 20 to 30 centimeter long piece of stem. Clear? 20 to 30 centimeter. Next, the plant in the natural position. So after the taking the small piece of stem which is 20 to 30 centimeter long, we treated with the help of GH, which is the growth hormone, growth promoting phytohormones in the plant, which is IB, indole butyric acid. Clear? As naturally it is called auxin. When we treat it with the root develop. Clear? And after then we transfer it into the soil. So this is the process by which we transfer the small cut piece of the plant stem in the soil for growing. In the last, the example of these process or the artificial vegetative propagation by stem cutting are sugarcane, grapes and rose. Clear? Again, artificial vegetative propagation by stem cutting. For a stem cutting, we take the small piece which is approximately 20 to 30 centimeter long. Clear? Then we treat it with the IBA to enhance the root development and then transfer it into the natural position. So plant it in the natural position in the soil. And examples are sugarcane, grapes and then rose. We can use these examples in case. Generally, stem cutting used in the herbaceous plant, especially the herbaceous plant. Clear? Now I demonstrate the type of stem cutting or the process of stem cutting in sugarcane. The sugarcane, the small piece 20 to 30 centimeter long cut piece of sugarcane. Clear? Now these cut piece contain the small buds. These are buds is the growing area or the node area. So it is the node and it is bud. It is essential area for development by the stem. Now we transfer these small piece of stem in the soil. Clear? Or after the treated with the IBA, then transfer in the soil. After few days, the bud area develop into the new plant and the nodal area now develop the new adventitious root. So we obtain the new sugarcane plant. Clear? So it is the process by which we obtain the new plant with the help of stem cutting. Now in the next process, it is the stem cutting. After the next process is by root cutting. And then by leaf cutting. So all are the same process. So we take the small piece of root, only the plant which capable to develop into the new plant by root. Clear? So we take the small piece of root, treated with the IBA, and then transfer in the soil so that the new plant develop. That time is the process is called root cutting. And the by leaf cutting, which is very rare cases. So a small piece of leaf and then treated with the IBA and then transfer it in the soil and produce the new plant. So it is the three type of cutting, which is the stem cutting, root cutting, and then leaf cutting. Root cutting and then other cutting generally used in the different type of plant. So it is the first type. Now we take the second type. Second type of artificial vegetative reproduction. The second type is layering. The type of layering means in which the plant part, the small part of plant or the small branch of plant bend and then transfer in the soil and develop into the new plant. Very simple process. We can take example of layering. One branch and then second branch. It is we can take the one branch and then root, and it is the second branch of plant. Now in the second process, the first branch at the same position and then second branch bend and then transfer and then second branch bend and then transfer into the soil. The part which is in the soil is injured by some instruments so that these part develop root, and after few days these plant develop the shoot system. That time we cut this part from the parental plant. We obtain the new plant by the layering. Clear? So it is a method of layering. The types of layering, it is the two types. First is called air layering and second is called mound layering. So we take the first, which is called mound layering. Mound. So a layering is a two type. First is the mound layering, second is the air layering. First type, which is the mound layering, which I have explained already. In this case, it is the most common method for propagation. Second, it is used in the herbaceous plant. That means the small size plant which branches or a small stem can easily bend. And then example of these, jasmine, grapes and strawberry. Clear? So mound layering is a simple type in which the small branch is bent and then transfer into the soil and then new plant. It already explained. Second, which is a very rare, air layering. In the air layering, generally it is used in the shrubs and the tree. That means the long size plant or tree in air layering generally used. In this plant, the stem part is generally not contact in the earth. Clear? That time we use air layering. Clear? So in the air layering, we select the branch for air layering. Clear? Now we cut the upper layer of these part. Clear? Especially the bark we cut it, and then we implant the mosaic or the grafting clay. Then we cover it by the polythene. So these covered by the polythene, clear, and the one water system is transferred at the area. It is called the gutti, the area where we develop the new branches, new stem. The area is called gutti, and the process is called air layering, because we have to implant it layering in the air. That time it is called air layering. It is generally used in shrubs and tree. Example, orange, lemon and lychee are the example of air layering. So it is a process by which we produce a gutti. After a few days, these gutti develop the root, and then we cut these part and then transfer into the soil and obtain the new plant of orange, lemon and lychee. It is the most common use in these type of fruit. So it is a vegetative reproduction by the layering. The third method, which is grafting. The next topic of the plant transfer into the second part of plant is called grafting. In this grafting, the two terms used generally. First is called scion and second is called stalk. The lower part of plant generally is called stalk and upper part is called scion. That means the lower part and then upper part completely develop into the new plant. So some essential requirement of the grafting. First requirement is that the both part cambium should be matched. Clear? So we cut both part. Both part means the scion and then stalk at the area where the where the cambium should be matched. It is the first reason. Second, they may be near about similar variety or similar species. So the lower stalk part fit on the upper part by this manner, and then tag by the tap and any other thread pieces, so it convert into the plant. After a few days it join at the area of cambium and develop into the new plant. It is called grafting. And next is the types of grafting. Types of grafting is whip grafting or tongue grafting, then vase grafting, crown grafting and bud grafting, in which the different angle of cut of scion and stalk takes place. So it is divided into the 4 different types of grafting. So these grafting generally used to improve the variety in case of plant, in case of vegetables and then fruit. So it is grafting. Our next topic will be, so next type of vegetable propagation, which is propagation of plant by the tissue culture or micro propagation. It is micro propagation. Micro propagation because we use the small part of plant. Small part means cell, then tissue, then seed, embryo. The small piece of plant now transfer into the tissue culture method, in which it convert into the undifferentiated cells cluster, or we can say the cluster of undifferentiated cell, which is called callus. Now these callus transfer into the different test tube and each test tube is treated with the culture media. That time the culture media means the nutrient medium, which is consist by the minerals and phytohormones and all required material for development in plant. So the small pieces of culture is transferred into the small test tube. After then few days the small plant develop, which is called plantlet. Now these plantlet is transferred into the small pot in the garden for hardening, and then it ready to use. So it is the process by which new plant develop is called propagation by plant tissue culture or micro propagation. So in micropropagation, we can produce thousands of plant in a small piece of part. That's why it is called micropropagation. Clear? So it is complete terms or technology with example of vegetative propagation. In our next topic, the definition as per I explained in last class, the process by which organism produce new ones without the help of reproductive organs and then reproductive cells is called asexual mode of reproduction. Clear? Next topic we will clear about the mode of asexual reproduction. Now five different modes of asexual reproduction. One is the fission, sporulation, fragmentation, budding and then regeneration. The five different types of mode of asexual reproduction which we will discuss. First is a fission, second is a sporulation, third is a fragmentation, and then fourth is regeneration, and fifth is a budding. Clear? After then we will discuss about the example of these types. First is the fission, two types, binary fission and then multiple fission. It is a two type. First is the binary fission and the second is the multiple fission. So two type of fission, first binary and second multiple. Now example of binary fission, protozoans, especially protistas, amoeba, paramecium and then planaria are the example of binary fission. Bacteria also example which is moneron. So it is example of binary fission. Example of multiple fission, amoeba and then plasmodium. Clear? Example of sporulation or spore formation, rhizopus, mucor and then albugo. Next, example of fragmentation which is pyrogyra. And then example of regeneration, hydra and then planaria. Next example of budding is a hydra and yeast. So these all are the just a scenario and outline of the mode of asexual reproduction. Clear? Now we will take one by one with example and then process of mode of asexual reproduction. Clear? So again repeat these five types or mode of asexual reproduction which is fission, sporulation, fragmentation, regeneration and then budding. Now I will take the first type which is fission. So first mode of reproduction which is fission. First of all, definition. The process by which organism is split into two or more than two halves and each half develop into the new progeny, that time it is called fission. It is of two types. First is binary and second type is multiple. Clear? So two type of fission as concerned of asexual reproduction. First is a binary fission, second is a multiple fission. Now first we take the binary fission. Binary fission means the word bi means two. So organism divide or split into the two equal half and equal half develop into the new progeny. That time the type of asexual mode of reproduction is called binary fission. The example of binary fissions are bacteria, the second is amoeba and third is paramecium and fourth is the planaria. These are the four different types of example of binary fission which I will already explain in the chart. Bacteria, amoeba, paramecium and then planaria. We will take only one example for the process or mode of reproduction. So process or mode of binary fission. Here we can take example of amoeba. So in case of amoeba, the binary fission is generally used by following process. Clear? So I will explain in very easy manner mode of asexual reproduction in amoeba. First we know that amoeba is protista, which is unicellular eukaryotes. And as you know, all eukaryotes possess the nucleus, well developed genetic material. Clear? So we can take the diagram of amoeba and then it is nucleus. Clear? It is a diagram of amoeba. Now, being the time of favorable condition, the fission is depend on the favorable condition. That means optimum temperature, then light, and then food. That time, amoeba undergo in the binary fission. So first of all, the amoeba and the nucleus, the division by amitosis. That means the nucleus divide without the division of chromosome. That type of division is called amitosis. So in which it is the amoeba, nucleus enlarge and in third step it is split into two nucleus. Now many events takes place during the time of second and then fourth phase, and between the second and third phase, sorry. So in second phase, the first, the nuclear enlargement, after then the cleavage for row formation, that means invagination of plasma membrane, and also the distribution of cell organelles. Clear? That means they have the cell organelles. So three events takes place. First, enlargement of nucleus. Second, invagination of plasma membrane, and then distribution of cell organelles. After the invagination completed, that means the plasma membrane invaginate and then fuse inwardly. That time it dissociate and convert into the two daughter amoeba. That time it convert into the two daughter amoeba. So these invagination now fuse and then dissociate at the site, and after dissociation the parental amoeba divide into the two daughter amoeba. Clear? So it is called binary fission. So it is called mode or process of binary fission in amoeba. Exactly same process followed by the bacteria, exactly same process followed by the paramecium and then planaria and other protista for increase the number. So it is a one type. Clear? Now second, multiple fission. So second is multiple fission. Multiple means more copies. In binary fission only two copies. So first, the definition of multiple fission. The process by which organism is split into more than two equal halves and each half develop into the new progeny. That time it is called multiple fission. Clear? Next is the process and then example. Example of multiple fission, which is plasmodium. Clear? That is also the protozoan. Unicellular eukaryotes contain nucleus. Clear? It is a plasmodium. Plasmodium falciparum, which is malaria causing organism. It divided asexually in human blood. So asexual reproduction in plasmodium. Also we can take example of amoeba by multiple fission in unfavorable condition. So in this case the plasmodium single nucleus divide many nucleus and each nucleus covered by the membrane, which is division or distribution of cytoplasm, and after then plasmodium burst and release all minute or tiny structures into human blood. During the time of malaria infection, these tiny structures are called cyst. Clear? A different name proposed in the case of plasmodium in human being. So it is a mode of reproduction, especially by multiple fission in case of plasmodium. Clear? So multiple copy produce and then multiple copy now release into the environment where they grow into the new plasmodium. So it is the second type of fission. Now we take the second type of mode of asexual reproduction. So second process is called budding. It is a mode of reproduction, especially asexual reproduction. Example is hydra. Clear? So first of all, definition of budding: the process by which new one produced by the outgrowth of animal body. The process is called budding. Clear? We can take the process of budding. Example, hydra. It is the hydra body with the tentacles. Now during the time of budding, outgrowth develop from hydra body. The outgrowth is called, it is outgrowth, and outgrowth is called bud. Now in next stage, after attaining the maturity by bud, that means maturity means the same size as the parent, the bud is detached from parental body. The bud is detached from parental body and develop as the hydra. Clear? So by this process, the hydra develop into the new hydra, and process is called budding. Now in next stage or next type, the third, which is fragmentation. Now in case of fragmentation, onset of maturation, first of all, it is example of fragmentation is spirogyra. Onset of maturation, the spirogyra body which is filamentous body, these filamentous body, onset of maturation, first and main requirement, onset of maturation. After the onset of maturation, the spirogyra body divide into two or more than two fragments, and each fragments develop into the new spirogyra. So it is daughter spirogyra. Clear? So the process is called fragmentation. Clear? Next types. After fragmentation, the fourth type is called sporulation. or spore formation. As we know the example or definition of sporulation, the process by which organism produces spores and spore develop into the new offspring. That time the process is called sporulation or spore formation. The example is rhizopus. The sporangium of rhizopus contain the nucleus which divide into many nucleus, and then each nucleus covered by the membrane, and now each nucleus is called spore. After the maturation of sporangium, these sporangium burst and then release the spores into the environment. When these spores land on any object and then develop into the new spores or new rhizopus plant. So it is a type of fourth type of mode of asexual reproduction, which is sporulation or spore formation. Next type, fifth, which is regeneration. First of all, definition about the concern of reproduction, the process by which organism produce new ones or new offspring by the cut body part. That time it is called regeneration. We can take example of planaria. If we cut the planaria into 3 small pieces, now each piece of planaria develop into the new planaria. So it is a small 3 pieces. Now during the time of regeneration, these small 3 pieces develop the remaining part and then develop into the 3 new planaria. So it is a planaria first, second, and then 3. So this type of reproduction, especially asexual reproduction, is called regeneration. So it is the 5 different mode of asexual reproduction, and the types, and then process, and then example. All mode of asexual reproduction performed by the single parents. One thing. Second, no genetic mixing takes place. Third, they are unable, especially so no genetic mixing in case of all asexual reproduction. So dear student, okay, thank you for watching my class.